So in our discussion of action potentials, we've used neurons. And neurons are the cells that undergo action potentials. So just a little bit about the neuron. You probably have had this in some anatomy classes in high school or around the university. But when we look at a neuron, there are going to be a couple of important parts. We're going to have the cell body, which is called the soma. And then we have processes that extend from that cell body. Many of these smaller, more numerous processes are called dendrites. The dendrites have information coming to the cell. But one long process that extends away from the cell body is called the axon, and that axon is going to transmit the signal away from the cell body and to a target. That target may be another neuron, it may be a muscle, it may be a gland. So dendrites bring in the information, axons send out the information. And since we have this huge interconnected network of neurons, and neurons interact with their targets via signals other than chemical signals, we're going to have all these junctions, neuron-neuron junctions and neuron-muscular junctions. And where they meet, that's called a synapse. There's actually going to be a physical space between the end of our dendrites, the end of our axons and the target. And it's that physical space where we're going to have to use chemicals to communicate. The electrical signal can't just arc from the neuron to the target. So dendrites are going to receive these chemical signals. And these chemical signals are going to bind to receptors that cause a change in our membrane potential. This is where we could have a positive increase or a negative decrease in our membrane potential. And we're either going to begin to refer to these as excitatory if they're more positive or inhibitory if they're more negative. That's what you do exclusively in physiology. So we can have an excitatory stimulus or an inhibitory stimulus. And all those changes of ions percolate around inside of the cell body. And whatever those ions and that change of our concentration of positives and negatives, whatever our membrane potential is at the beginning of the axon, called the axon hillock, where we have a huge concentration of sodium voltage-gated channels, that's what determines if we send the signal down the axon or not. Our dendrites and our cell body, they're going to have few, if any, sodium voltage-gated channels. So we're not going to start the action potential in the dendrite or the cell body. It's going to happen at the beginning of the axon. And the axon then sends that wave, like we saw in that first illustration, all the way down the axon to the very end. Now, do you recall how rapidly we said we could send that signal? I mentioned it earlier. Over 100 meters per second. Now, just a normal axon by itself can only transmit a signal about 1 meter per second, which is still pretty impressive for a cell. So the way we increase the signal of our action potential moving down an axon is we insulate it. I think of it like electrical tape. It's basically insulating cells, Schwann cells for our peripheral neurons, oligodendrocytes in our central nervous system. They wrap their plasma membrane around the axon maybe 40 or 50 times. Just like if you've ever seen an electrician with a roll of electrical tape, they start it and then they just wind it. And the more layers, the thicker that gets, the better the insulating function of our myelin, which is basically just phospholipid layer after phospholipid layer. No cytoplasm between. But when we wrap sections of our axons, it's not going to be one continual layer of insulation. They're going to be patches of myelin with space between where the axon is exposed, and those gaps are called nodes of Ranvier. Now, under this myelin, under the insulated layers, we don't have sodium voltage gated channels. We don't have potassium voltage gated channels. We don't have sodium potassium pumps because nothing's going to get through. It's a watertight barrier, essentially. And so what happens is our action potential is beginning at the axon hillock, and as it's moving down our axon, it's going to appear to jump from node to node to node. And this jumping and the moving of ions is what speeds up that transmission of the action potential over 100-fold. That's what makes it faster. This used to be called saltatory conduction, skipping, because that's the conceptual idea. But it just increases the flow of our ions, because the sodium that comes in at this node, when that sodium comes in, it spreads in all directions. So that sodium coming in changes the membrane potential at the next node. And this node will reach minus 55. The sodium voltage gated channels here open. Sodium comes in, spreads in all direction. So the sodium coming in here is going to cause the opening of the sodium channels here. Do you see how it just, once you start it, it's not going to stop. Because each sodium voltage gated channel will be responsible for opening the next and the next and the next, all the way to the end. And so here we have on the left an unmyelinated axon. And this is just an illustration, but it shows the transmission of the signal. You see how slow that is? But once you wrap the myelin around the axon, now look at the transmission. It just zips along. And so the effect of myelin is to drastically increase the transmission of your electrical signal, that action potential. Now, this sort of gets at a question. Can you live without myelin? And in fact, the answer is yes, but your quality of life is not going to be as good, because that's multiple sclerosis. Multiple sclerosis is a degenerative disorder. In fact, most of the time it is autoimmune. Your own body attacks the cells that produce the myelin, and you lose those myelin sheaths. And so you're not stopping the signal. You're still sending the signal, but you see how it's really slowed down, so the coordination is totally off. And if you've seen someone with multiple sclerosis, even the simplest of tasks like walking, everything's slowed down. They're being much more careful in everything they do. So here we are, pardon me. Here we are at the end of our axon. We have this synapse. So we need to change our electrical signal from our neurons into a chemical signal that's going to diffuse across this short space, about 20 nanometer space, to be detected by our target cell. Again, gland, muscle. And so what we're going to see is that chemical signal are referred to collectively as neurotransmitters. Neuro from nerve, transmitter, meaning transmitting this signal. So here we have in our illustration an axon. There's our action potential coming down. We have our axon here at the end. You can see this space. And in this case, we're saying, okay, this is between the axon of one neuron and the dendrite of another neuron. And that electrical signal is going to be detected and cause the release. What process would we call this? Here's a little vesicle with our neurotransmitter. Fuses with the membrane, released. Exocytosis. Secretion is a form of exocytosis. But our action potential causes the release of all these chemicals into this space. And because of diffusion, we got a high concentration here, low concentration at the target. Those chemicals diffuse very rapidly across that short space and are detected by the receptors on the target cell that then either directly or indirectly can lead to the generation of a new action potential in that next cell, as strong and as powerful and as rapid as the one that just got there. Hit the end. The action potential stopped because we got to the end of that one. And that's it. And action potentials only go in one direction too. Why do action potentials only go in one direction? Correct. It can't go backwards because the sodium voltage gated channel that just sent you the signals in the refractory period. So it's one direction all the way to the end and then just stops because it can't go back. So with our neurotransmitters, again, we're talking about these chemical messengers, largely protein in nature or other chemicals that are derived from amino acids, for instance. They're going to be diffusible, releasable, and they're going to elicit a response in the target. That's sort of a generalized criteria for these things. And they're going to be detected by receptors. So once that chemical binds to its receptor, it can either be excitatory. Now, I think I may have changed the wording since your PowerPoint, so if you've got an old PowerPoint, make sure to put this in. An excitatory receptor, when a neurotransmitter binds to it, it's going to lead to a positive change in our membrane potential. Some neurotransmitters, when they bind, can be inhibitory. And that's going to cause our membrane potential to become more negative than resting membrane potential. So excitatory, more positive, inhibitory, more negative. This is going to make it easier to reach our threshold for an action potential, hence excitatory. This is going to make it more difficult to reach that threshold because we've got further to go to get to minus 55. Now, just a few neurotransmitters to talk about. The most common is acetylcholine. One of the reasons it's the most common is because it's the neurotransmitter that signals from a motor neuron to a skeletal muscle. And you got a lot of skeletal muscles. Acetylcholine is also used in some neuron-neuron synapses, especially in your parasympathetic nervous system, this rest and digest system. And acetylcholine is always excitatory. Why would acetylcholine necessarily always be excitatory if you're going to your muscles? Wouldn't you need your muscles to be able to relax? So shouldn't it be inhibitory? How do you inhibit your muscles? Stop telling them to contract, right? So that's why acetylcholine, it's just the on switch. The off switch is no acetylcholine. So that's how it can always be excitatory. Whenever you talk about synapses or neurons that secrete acetylcholine, the adjective you use is cholinergic. See where we get choline, acetylcholine? You put ergic on the end of a word in biological terms. That's referring to that type of. So a cholinergic neuron is a neuron that secretes acetylcholine. A cholinergic synapse is a synapse in which you use acetylcholine. Okay? The junction between a neuron and the target where there's a little space, where you're going to have to use these chemical neurotransmitters. Another broad category of neurotransmitter are referred to as the catecholamines. Catecholamines are neurotransmitters derived from tyrosine, a single amino acid. And catecholamines, one example of catecholamine is norepinephrine. Now, epinephrine is a type of catecholamine, but epinephrine is a hormone. It's produced in the adrenal gland. Norepinephrine is produced by neurons, and so we say norepinephrine is a neuron, or a neurotransmitter, sorry. And so if neurons are producing norepinephrine, then we say those neurons are adrenergic. Now that may not immediately be logical. Cholinergic, yeah, produce acetylcholine. We got the names the same, right? How do we get adrenergic from norepinephrine? The adrenal gland. Another name for epinephrine is adrenaline because it's produced in the adrenal gland. epinephrine, epi means above, nephros means kidney. That's the location of the suprarenal glands, the adrenal glands. So it all relates to that, but sort of in an anti-intuitive sense. But catecholamines are important in the central nervous system. They're important in neurons, between neurons and your smooth muscles, particularly in our fight and flight response. We're going to see a lot of norepinephrine used in our fight and flight responses. So those are two broad categories of neurotransmitters, but they're both going to be regulated. The release of these neurotransmitters from the axon is regulated largely by calcium. You remember in one of the previous slides, we saw the exocytosis that's happening? That exocytosis occurs because as our action potential is sliding down the axon, when it gets to the end, it's also going to open voltage-gated calcium channels. We're going to see calcium is a potent activator of second messenger signaling, which we might touch on it today, but we're going to see most of that on Monday. Second messenger signaling, it leads to a cascade resulting in the exocytosis of our neurotransmitters. So calcium, it's not just for bones. You've got to have it for nerves to signal. You also are going to need it for muscles to contract. You've got to have calcium in your body. So here's another illustration. Action potentials coming down. Calcium voltage-gated channels opening. Calcium comes in. This shows just a little bit of the second messenger signaling that happens, leading to the docking, fusion, and exocytosis of our neurotransmitters. These details aren't critical right now. The critical part is calcium comes in. Calcium doesn't come in. You don't get the exocytosis. You don't transmit the signal. Your neurons have to have calcium for that release. So that was the axon and the neurotransmitter. Now on the target, you have to have a receptor. And for our skeletal muscles, our acetylcholine receptor, our cholinergic receptor, is referred to as a nicotinic acetylcholine receptor. It can also be stimulated by nicotine. That's how it got its name. But you have to bind 2 molecules of acetylcholine to the receptor. And when we talk about receptor and the chemical interaction, we refer to the acetylcholine as the ligand. So this is either a ligand-gated or a chemically regulated receptor. But what's cool about the acetylcholine receptor is it's also an ion channel. So when 2 molecules of acetylcholine bind, it opens up. Guess what comes in when you open this nicotinic receptor? Sodium comes in. Does this sound familiar? That's what's happening with the action potential, right? As sodium comes in, it increases our membrane potential to, guess what? Minus 55. Guess who's next door to the nicotinic receptor? Sodium voltage-gated channel. So when you open the nicotinic receptor because of the acetylcholine, that leads to the voltage change that automatically opens the sodium voltage-gated channel. And what have you just restarted when you open a sodium voltage-gated channel? The depolarization and action potential. So you've taken an electrical signal from the neuron, converted it into a chemical signal. Now you've transduced it again back to electrical signal. Takes off. Because the electrical signal can't cross the space, but chemicals can by diffusion. So here's an example of our nicotinic receptor. Notice it's closed. Why is this one closed? It's only binding 1 acetylcholine. But when you bind 2, there comes sodium. It comes rushing in. It does allow some potassium to go out, but sodium in is the principal one that leads to this excitatory positive increase in our membrane potential that leads to automatically an action potential and contraction of our skeletal muscle. Now, we have this other neurotransmitter, GABA. We don't have to worry about, I think it's like gamma aminobutyric acid. That's for physiology. Just know another transmitter that works in a different way. In this case, when we bind GABA to this receptor, it's a chloride channel. And when the chloride channel opens, that's going to lead to a more negative potential because chloride's going to come in. We're going to hyperpolarize. So that's going to be an inhibitory type of reaction. So do you see how it depends on the receptor and it depends on the ion if we're going to be excitatory or if we're going to be inhibitory? Correct. Below resting potential. Now, have you seen a problem yet? Action potential's coming down. We secrete all of these chemicals into this little bitty space. Let me give you sort of a preview of how many of these molecules there are. In a generalized acetylcholine release in a neuromuscular junction for skeletal muscle, there may be upwards of 30,000 vesicles that release their neurotransmitter. And each vesicle may contain 10,000 molecules. That's a lot of zeros, people. All of that flooding into that synaptic cleft, binding to the receptor. How do you stop it? How do you stop the signaling? With all that chemical around, it seems like you'd be contracting for a long time, right? The inactivation of the neurotransmitter happens as fast as the signal gets there to start with. And on skeletal muscle, it happens because you've got an enzyme on the membrane of the muscle called acetylcholine esterase that destroys acetylcholine. Which to me makes no sense whatsoever. You're making 10,000 times 30,000 molecules only to get over there and be destroyed. Why in the world would you make so much only to break it down so fast? The way you speed up diffusion, increase the concentration gradient, flood that space with a ton of chemical. It's going to get there fast because you want the signal to transmit fast. But you want it to stop fast too, so as soon as it gets there and sends a signal, you want it to stop, so you eat it up with the enzyme. In other systems with other neurotransmitters like the adrenergic, the norepinephrine, reuptake happens. As soon as the cell secretes all of the neurotransmitter, it turns on neurotransmitter vacuums and sucks it back into the cell. Again, we've talked before about, y'all grew up with smartphones and computers and stuff. When we were kids, we had jumping beans and a little paddle ball. A little red ball with a rubber string. You hit it, it goes out and it comes back. Well, that's what the cells do. They throw out the neurotransmitter and just bring it right back. That's the reuptake that happens. So here in this illustration, we have norepinephrine. So this is an adrenergic neuron. It's secreting the norepinephrine. Here's our receptor. But most, I think upwards of 70-75% of the neurotransmitter is sucked right back up into the cell and deactivated and recycled to make more. You got to have a ton to send the signal fast, but you got to have a way to stop the signal so that you can do it again. Endocytosis, correct. So exo-endo is just rapid.
You said it was adrenergic.
Adrenergic. If it secretes norepinephrine, then that means it's an adrenergic neuron.
So